The last speaker is Guy Cochrane. He's leading the European Nucleotide Archive of the European Bioinformatics Institute at the Wellcome Genome Campus of Cambridge, UK. He has set up in 2019 a platform for data storage for pre-publication sharing of pathogen sequencing data. And in the COVID-19 context, the European Bioinformatics Institute has used its infrastructure, expertise, and network of international collaboration to build the so-called European COVID-19 data platform. And this platform has integrated a lot of COVID-19 biomolecular data. So, Guy, please, you have the floor.
So thanks very much, Eric, and thanks to ICPerMed for the opportunity to present and to discuss. So indeed, this is a presentation about a data platform that really aims to provide a foundation, an evidence foundation for the response to COVID. That's about providing mechanistic information, biological information about virus, infection process, transmission, spread, and so on. But also, of course, as one looks for more targeted and more personalized approaches to medicine, of course, it's the evidence basis for this as well. So what I'll do is I'll just introduce the platform quickly. I only have time to cover a few areas. I'll cover 3 specific areas of development, and I'll touch on a few other areas at the very end. So the platform is part of the European Union's Era versus Corona action plan. So this is the major scientific data part of that approach to COVID. We launched very early in April last year, and at the time we were already able to put together quite a significant data set. On the right here you see the website that is the entry point for most people, but that's only one part of the system. Since that time there's been a lot of energy, and we've been doing many different things. So I really can only cover a few of those in a few minutes here. So the focus is on biomolecular data, so data that relate to biological molecules and information carried in those molecules. This concerns the virus, patients infected by the virus, but also to make sense of this, one needs the context from therapeutics, diagnostics, so the clinic, information about contacts and exposures, but even going further out, we've heard about the importance, for example, of socioeconomic factors, so data there, social science data, epidemiology, and so on. So a center point on biomolecular data, but contextualized. So to build such a system, the implementation requires multiple parts, so multiple parts in multiple geographies, effectively. So on the top left here, my institution, the EBI, we're able to centralize many of the biomolecular data, particularly those that can be made openly available. For sensitive patient-linked data, including the biomolecular data, but also clinical and diagnostics data and so on, these tend to remain within the country in which they're produced, safeguarded within the jurisdiction and the privacy regime of the country's health infrastructure, or wherever it is. So there we need bilateral connections to be able to connect across these different geographies. We also have connections into the larger European level research infrastructures, for example, around compound screening, clinical trials, animal models, imaging, and so on. And then many different areas have international data exchange, data standard connections as well. So we're generously funded by the European Union across 5 different Horizon 2020 projects. The partners are EBI, Elixir, which is the European network for biological data, the European Commission itself, and a host of institutions in the public health, clinical and academic areas and technology areas that we worked with for some years on infectious disease in general. So one of the first priorities really was to make available data quickly and in an integrated way for people to use. And we did this across different platforms. And so here if you look at the bottom of this first, you see the different types of data. I'm going to talk about viral sequence in a moment. We also have host sequences. We've heard a little bit about host genetics already. Association studies, expression studies, data from proteomics platforms, structural biology platforms, metabolomics, imaging platforms, and of course the very rapidly growing literature. So essentially there's a website and a series of interfaces that one can go in and find in a joined up way different data that relate to COVID. Of course, many of these data are reference data that pre-existed COVID, For example, we have coronavirus data from things other than SARS-CoV-1. We have data from animal hosts of those coronaviruses. And of course, we know a lot about human genes and proteins and so on. But over time, we've been able to add these quickly, and we will be adding more. There's now an emerging network of national data portals to connect into the central system as well. And the system's being nicely used. Here's an example of how one group approached it and used it. This is the Open Targets Initiative. And they came in and they pulled data that were from the portal. They put together essentially a catalogue of potential targets, so genomic targets, and against those they have, through analysis and curation processes, they've organised and annotated compounds that have potential activity against those targets, and they've provided details of the trials that they may be involved in and so on, and then links in many cases, to functions and impact of changes in these targets and so on. So the next element is the virus itself, and of course, part of the exposure, as we heard, is that's important in response is likely to be the genetic variation in the virus itself. And so from the start, we prioritized the mobilization of data. So as countries set up sequencing activities for the virus, we really pushed that the data would become available openly for people to reuse, really to understand deep variation and to separate true signal from noise. All sequencing is, of course, erroneous, or all sequencing has a certain rate of error that can't be ignored. One needs systematically to process and to understand and treat error in a particular way, so that one can call true variation. So we sought to mobilize raw data, and we provide a thing we call the SARS-CoV-2 data hub. So it's essentially a toolbox for people who are working with viral sequencing data to manage those data, to share the data, to run analysis, so there are variation calling tools, assembly tools, annotation tools, phylogenetics, and tools to explore all of this. So we made this available here. On the left, for example, this is a thing you use to upload your viral data into the system. This is a visualization of some of the QC for assembly data, I think. And here we have some phylogenetics. So there are different tools that are on offer. So we have now over 300,000 different viral isolates represented in raw data from 62 countries. And this platform has actually led the mobilization of most of the data that are available to the world. So this has been quite pleasing to see. Of course, there's much yet to come. And the system's being used both by many different external users, but we've also started systematic analysis, such as in variation calling ourselves. Here's an example of usage here. So this is the Centre for Genomic Regulation in Barcelona. So using the data mobilized from the virus through the data hubs and the data portal, this is a tool that they pulled data and they ran variation calling systematically. And this is a tool that allows one to investigate individual variations, looking at the frequency, the occurrence, and the samples, the distribution of those occurrences, and then mapping them to positions on genomes, and then perhaps going on to look at the function of those particular regions. So the third component I'd like to mention relates to the sensitive human data, so patient level data. The focus here has been really on sequences, particularly on host genetics and the sequencing of response, say transcriptomes in response. But the system is actually suitable for other data types as well. So what we're doing is we're building on an existing piece of infrastructure, the European Genome Archive, which is able to handle control in a controlled access way, securely retaining privacy, data at patient level, particularly sequence data. And the work here is to build this out into a federation. And the idea is that each country can have a node of the database, and they can continue to store the data from the patients within the country within their own system, behind their own firewalls, but present systematic interfaces that allow a federation to be created across all of these national databases. And so a single accessioning system, a search service that can run from anywhere to find the data sets of interest, regardless of where they are. But also when one has to make an access request to see a particular data set, the same way of doing that regardless of the location of the data, and then if access is granted, the same interfaces for engaging with and computing on the data when there is access. So this is something that of course takes some time, but we have 5 nodes across Europe that are working hard at this with technical implementations already. Testing is going on. and currently there are legal agreements and governance issues to be resolved. But of course, while it's happening, we do have the central system, and for those that can use it, it's being used, and we are gathering host genetic studies into that system. I've just put this in here. So Jack mentioned the Host Genetics Initiative. One of the, if you're part of this, your data go to this EGA system. So it's all sort of connected with these big public initiatives. So finally, just time to mention a few other things. This is certainly not everything, but some of the things that are going on. We're building a system that will better allow the integration of studies from different cohorts at the level of the cohorts to be able to explore and discover and perhaps connect across different cohorts. We're working on different data standards. In some cases, there is little sharing of certain data types. So serology is an example of that. Where we're seeking to work with the community to develop the appropriate standards and systems for sharing the data. This is an example from the Netherlands, where there are some very nice deep data sets where you have viral sequencing, deep viral sequencing, you have laboratory results of deep profiles of B cells and T cells, and so on. You have clinical data as well, very deep and rich, but very fragmented in their current layout. And so we're using the platform there to try to tie these together, and this is a pilot from the Netherlands. But the power of being able to compute and ask questions across these sets, these data sets, is great. This is an indexing system to bring in external data sources. There's a lot of work on analysis development tools, and of course on data curation and quality control in different parts of the system. So I leave it there. I thank you for your attention, and I look forward to the discussion later on. Thank you.
Well, thank you very much. Very impressive organization and infrastructure, indeed.